August 1st, 1920. A girl named Loretta Pleasant is born in Clover, Virginia, tobacco country. History would eventually know her as Henrietta. She grew up in a small cabin that once housed enslaved ancestors. She laughed easily, wore red nail polish, loved to dance, and raised 5 children. In the 1940s, she and her family moved to Turner Station, Maryland. It was a working-class black community anchored by heavy industry, where her husband took a job at Bethlehem Steel. Henrietta's story started in the fields of clover, rooted in family and the daily work of survival. In 1951, human cellular research was stalled. Scientists were unable to keep human cells alive outside the body for more than a few days, making it impossible to observe the long-term effects of drugs or disease. That same year, Henrietta felt a knot of unexplained pain. Because hospitals were heavily segregated, her options were limited. She sought help at one of the few institutions that treated black patients, Johns Hopkins. Doctors examined her and delivered a grim diagnosis. She had an aggressive, unforgiving tumor on her cervix. During her radiation treatment, doctors extracted 2 samples of her tissue. She was never asked. She never gave consent. This silent extraction enabled decades of medical research, even as it bypassed the fundamental requirement of patient consent. While one sample died, the other did something no scientist had ever witnessed. The cells did not stop. They divided, and kept dividing. Researchers named them using the first 2 letters of her first and last name, HeLa. They had discovered the world's first immortal human cell line. For the first time, researchers had a stable, permanent testing ground for human biology. They could expose these cells to toxins, radiation, and infections without risking a living patient. Mass production began immediately. HeLa cells were packed into vials and shipped out to research centers across the globe. Their first major global contribution provided the testing data needed to develop the polio vaccine, leading to mass inoculations that saved millions from paralysis. HeLa cells allowed scientists to observe exactly how viruses interacted with human DNA. This led to the development of viral vectors, hollowed-out viruses used to deliver helpful genetic material directly into the body. HeLa cells shifted the focus of modern medicine from observing human illness to engineering human immunity. In the 1960s, NASA needed to understand the effects of zero gravity on human biology. Before sending human astronauts, they launched HeLa cells into space. The cells survived. In fact, they divided even faster in zero gravity, proving that human life could survive and adapt beyond Earth. But while her cells were orbiting the planet, Henrietta's body had long been buried. She died in 1951, at age 31. Resting in an unmarked grave. Her cells generated billions of dollars in research value. Meanwhile, her 5 children lived in Turner Station, unaware of their mother's cellular legacy and often struggling to access basic medical care. This era of scientific discovery was built upon a foundation of extraction, silence, and systemic neglect. During the Human Genome Project, scientists relied on HeLa again. By fusing Henrietta's cells with animal cells, they could map exactly where specific genes live on our chromosomes. That map eventually met a tool called CRISPR-Cas9. It acts as a molecular pair of scissors, allowing scientists to precisely cut and edit DNA. To prove this tool was safe for human therapy, scientists used HeLa cells as their initial training ground, ensuring CRISPR could make precise cuts without causing unintended cellular damage. This medical requisition form represents a modern breakthrough. Scientists are using CRISPR to reverse the mutation that causes sickle cell anemia. Looking at this blue vial, you are seeing the culmination of that process. Doctors extract stem cells, edit them, and reintroduce healthy cells. The ability to cure a generational genetic disease is linked directly to the decades of validation performed on HeLa cells. Henrietta Lacks' contribution is woven into modern survival, from the fight against HIV and COVID-19 to the development of the HPV vaccine, which prevents the very cancer that took her life. Without her cells, medical research would be significantly delayed, and millions of lives would have been lost to preventable disease. Acknowledging her story is more than a historical correction. It places the truth of her life back at the center of human survival. Say her name. Understand the immense weight of what it carries. As long as science advances and humanity reaches outward, Henrietta Lacks is still in motion. She is alive inside the system.